Okay, welcome everybody. This is the final meeting of the NE1 from 2020. And today the program is we're going to go over the last photo exercise. We've been slaving over it, Rui and I guess maybe some of you. There's nobody that really completely solved this puzzle. I only got one entry from Yves. He now wins the pizza prize. So you need your address of the extra large pizza delivered to you. I'm going to go over, I spent many hours on this, like at least 6 hours. I tried to figure out this stupid mystery. I discovered multiple bugs in our own Python notebook. Anyway, we're going to spend a few minutes on that. I hope the exercise when it's instructive, because later on when you guys run Spice, just simulate your circuits, you realize it's really only doing the same thing. The only thing that Spice does is the models are much more complicated for the transistors. It includes all kinds of parasitic elements and parasitic capacitors between everything, all kinds of second order and third order effects of transistors. And the solvers, the numerical solvers, which you might have had a struggle actually with this solve IVP, getting the thing to solve. Trust me, the solvers in Spice are much more sophisticated than the solve IP, because they have to solve these huge systems of thousands of transistors where nodes are floating, and they have to get this thing to numerically be stable. So I hope you appreciate when you run some of these circuit simulation tools, what's really going on is just what you guys were doing here. The beautiful thing about doing it this way is that it's pure mathematics. You have to realize that what you've simulated here is not reality. It's just your model of the transistor. It's a very simplified model of the transistor. It's an ideal model of the transistor. And so if you can really understand it working with this ideal model, then you can really get a handle on the mathematics and stuff. And sometimes that's hidden by the parasitics that appear in reality. So that was the point. I think it's a cool exercise, personally, because it teaches you quite a bit about the limitations of such tools. And so there was this mystery that we had. Hi, Giacomo. The mystery was that when we calculate the Q equals 1/2 condition, do you remember this for the stable ringing response of the transimpedance photoreceptor? Remember that the tau2, the output time constant has to be smaller than tau1 by at least the tau1 divided by the loop gain. This is the gain going all the way around the photoreceptor. Remember, you have an A from the amplifier, and then you have a kappa going back through the source follower transistor. And there's a 1 there too that we left out before. If this condition is true, if they're equal, then you should have a perfectly stable step response at that particular photocurrent with this bias current that gives you this tau2. Okay. So so to go back to this, we had this Q. Do you remember this expression from the previous lecture? So first we're going to go over this thing, and then we're going to show you a whole bunch of demos. Retina, optical flow, cochlea, audio recognition with cochlea, and then winner-take-all neuron, multi-neuron spikes. But I thought I would just go over the lab exercise a bit first. So the stable response, Q equals 1/2, meant that you had to make the output time constant of the transimpedance photoreceptor smaller than the photodiode time constant by a factor of the loop gain, +1. And the problem is that when we tried to show that, we would continually get the condition that it wouldn't be stable. Instead of making a nice response that looks like this to the step, it would overshoot and ring a bunch of times. And we couldn't figure out why. Like, what's wrong with the theory? Kept going over the mathematics, discovered some mistakes in the ID the subthreshold current equation. Just couldn't figure it out. Finally, do you know what the solution was? What the result was? Yves kind of found it. So to get this Q equals 1/2 current, you need the gain. And how do you compute the gain of this amplifier? Well, we assumed that the gain of the amplifier was A here was equal to VE over UT, right? Kappa VE over UT. That's what we assumed the gain was. And why did we assume that gain for the amplifier? Because the model of the transistor was this. Here is the drain voltage. Here is the drain current. The model of the transistor is like an ideal saturation current. This is ID sat. Plus this drain current component, the early voltage component, where this intercepts back here at minus VE, where this GM here is equal to ID sat over the early voltage. So this is like a constant current part here, and this is the parallel conductance part. Okay, and that was mathematically how the subthreshold equation was defined in our notebook. It's this ID sat plus exactly this parallel conductance. At least that was how it was supposed to be. I'm going to show you the notebook, what this equation actually looks like. Here's the actual subthreshold equation after I finally got it correct. And this is after like 5 revisions. Now I finally broke it down. So here's the ID sub equation. It gives the drain current as a function of gate, source, and drain voltage with a particular thermal voltage, off current, and kappa, and early voltage. And before, there was actually an assumption that this current at which you compute the drain conductance is the actual current. It's not the saturation current. So now it explicitly computes the saturation current as I0 e to the kappa VG minus VS over UT. That's the constant current part, right? And then it says that the actual drain current without the drain conductance is ID sat times 1 minus e to the minus VDS over UT. And then it adds on, and there was a mistake in this equation in the original notebook. We thought we fixed it, and then there was another mistake. I finally fixed that. So now the actual drain current is the original drain current, the saturation current. We compute it right here, plus 1 plus VDS over VE. Is this correct? Do you think this is correct now? I think there's a mistake here still. You see what the mistake is? Giacomo, are you there?
I'm here. I'm trying to figure it out.
So this is the constant part of the drain current that we just computed. This ID, which is the saturation current times the drain, the subthreshold drain saturation part. And this is supposed to be the constant conductance part, the constant drain conductance. Should it be VDS or just VD here?
Just VD.
It should just be VD, right? Because the source has nothing to do with its channel length modulation. It's just a pure drain effect. Actually, the source is just at 0, but this is actually incorrect. Do you see that? So it should be this. It should just be the drain voltage, because it's the drain to bulk voltage that affects the channel length modulation. And at least that's a better approximation. Okay, anyway, so that's the equation. And then from that equation, you should get, be able to compute the amplifier gain is just this transconductance divided by the drain conductance. That's the ideal mathematical voltage gain of this amplifier in small signal. But in reality, is that what it is? So finally, after a long, long time of trying to puzzle out why the thing wasn't getting stable at Q equals a half, did a simulation. What is the actual open loop voltage gain of the amplifier in the photoreceptor, which is this little amplifier, very simple amplifier, just a constant current IB with this FET. So this is VG and this is VD. And remember, this is the ideal transistor. It has this parallel conductance, which is GD, which is ID sat over VE. Okay, so we should be able to simulate that, right? That's just a separate circuit that we can simulate. We can make a transient simulation of it, put a little tiny capacitor on here, any capacitor, and just do a time domain simulation. Sweep this voltage and look at this voltage. It should go down like that, right? As you cross the threshold. And so the result of that was, I'll show you. Can you see the notebook now? Go down and look at that simulation.
Yes, we can see it.
You can see it, right?
Mhm.
Okay, so here's our step down. We change it. So it's now just a downgoing step. It goes from the starting photocurrent down to one that's just a bit smaller. Small signals downward going step. See, it's just a small step, right? Like 10% step. Okay, that's the actual step response. Here is the simulation of that open loop amplifier. Here we compute the derivative of the output voltage with respect to time and the drain voltage. This is like the differential equation, right? The current flowing into the output node is the bias current minus the drain current. The drain current is just the subthreshold current with this particular kappa and VE. The gate voltage is computed inside this function. It's just a linear sweep starting from V0 and ending up at V1 at the end of this time TT. So the gate voltage is just sweeping up or down. And then what this thing computes is the derivative of the output voltage, which is the output current divided by this capacitance. Okay, so then this thing solves that from over the time. And then it plots the result. So here's the plot of the output voltage, V out in volts versus V in, VG in volts. So this goes from 0.4 to 0.43 volts. It only goes 30 millivolts. And this output voltage goes 17 volts. That's a really big voltage, right? Remember, there's no VDD, so the voltage can go up to like 1,000 volts here, right? So as the gate voltage gets higher and higher, of course, the output voltage goes down, right? As this gate voltage goes up and gets bigger than IB, then this output voltage will go down, right? If this saturation current gets bigger than IB, then of course this transistor, what happens to this transistor? The saturation current is bigger than the bias current. So what must the output voltage do? It must go smaller and smaller. Does it go all the way to ground? What happens when the voltage across this drain source gets really small? So the transistor goes out of saturation, right? So the drain voltage will get really small, like a few millivolts, until the voltage here, until the current flowing through this equals the bias current. So that's what you see in the simulation here. See, this is where the voltage here, it gets high enough that it's bigger than the bias current, and then the voltage here gets really small. But during this whole point, it's supposed to be just a line. It's supposed to be exactly just that conductance. But see, it's actually curved. That's because the transistor is still exponential. And so the actual gain of this thing depends on where you measure it. Now, I measured it here in a simulation between VDD over 3 and 2/3 of VDD. So this here picks out the range here. And so it's fitting a line right in this region here. And when you set the early voltage to 10 volts, so it's a nice long transistor, and you compare the slope of this line, which is 350, that's the gain, with the estimated voltage gain, 320, it's very close. But what happens if you do it for an early voltage of, say, 2 volts? Let's do that. What's going on here? It's not defined. I have to run everything above this.
I think that maybe in this thing before you did when you changed
Did I make a mistake?
from VDS to VD, you didn't put the underscore. And I think you defined it with the underscore in the definition of
in my subthreshold equation.
Yes.
Oh, thank you. Sorry, guys. Thank you. Okay, now let's just go back down here. going to run everything up to here. Across my fingers that it runs. Where is it? Is it here? Yeah. Is it finished already? It's still running. Okay. Okay. Now the measured gain is 92 and the predicted is 64. The actual measured gain of this inverting amplifier is much higher than the predicted gain. With this early voltage, the predicted gain is 64. In fact, it gets even, before it was a much bigger ratio. I've set the early voltage to 1, which I think was the default. Now the measured gain is 60, the predicted gain, and now it's twice as big as you predicted. So there's some nonlinear effect, that's going on. Even though everything should be perfectly linear, this little bit of exponential here is making the gain different than what you predict from theory. And that is enough to explain the fact that it was ringing, because you assumed that you knew what the amplifier gain was from theory. And it wasn't that way, even in simulation. So that's the main reason that it didn't work out before. All these effects are partly a result of the fact that when you make the early voltage small, then the loop gain is not much bigger than 1, right? Like if I set this, I haven't tried this, but I set it to like a really big number, like 30 volts, and run it. I'll see if it even works. Yeah, it starts getting into instability, but now you see it's closer and closer. It's like 670 here, and the predicted gain is 640. Just measuring it right here in this little voltage range. Okay, so I don't know. I hope that's kind of helps a bit. Okay, so let's go on then. Let me go back here. So do you have any questions? So the final drop dead day to upload your exercises for all the labs. I mean, if you have improved them, you're welcome to upload some improvement, is this Friday, the 18th. Okay. Okay. Before we start showing all these demos, I want to know, are people interested in preparation for the exam and just having some sessions where we can talk about circuits or ideas? You can ask questions, you can practice scribbling your answers and stuff like that. Are you interested in that? Because I can schedule some, say, at the same time every week. Nobody's going away anywhere. I could just schedule some time to scribble circuits with you guys if you want.
Yeah, that would be cool.
Okay, cool. Then you can just ask questions and we'll just scribble the answers and try to show you how to answer the exam questions. Cool.
Okay. Thank you.
Okay, so we're going to now do some demos for you, a bunch of neuromorphic stuff. So you already have a great preparation for this stuff because you've studied now basically the input circuit for a silicon retina. This whole exercise was just to study this input circuit, the one you've just simulated. And I'll just show it. The best way is just to show it that way. Can you see now the whiteboard?
Yes.
Okay, good. Okay, so you have that photoreceptor, right? I'll start with this photoreceptor. And there's light shining on it. And then there's an amplifier. Let's just draw it like an inverting amplifier for now. Okay, that's the thing you guys just simulated. Nice logarithmic photoreceptor, nice and fast, low noise. It's a good front end, right? Gives you a logarithmic voltage that changes with a photocurrent logarithmically. So you get big dynamic range, like a factor of a million in photocurrent. Easy to achieve, 120 dB. So now you'd like to make spikes out of it, like the eye sends to the brain. So how can you go about that? What kind of spikes can you make? I mean, you could maybe take the photocurrent itself and just put a neuron on it, and then send spikes out at a rate proportional to the light intensity. Does that make sense? Like if each pixel would just send spikes proportional to the light intensity, would that be useful? Not particularly, right? The problem with that is that really bright pixels would send tons of spikes and really black ones would take forever to send a spike. And so like the speed would vary with the brightness of the picture, you know? So you'd be spending all your bandwidth on pixels that are brightly illuminated. That might be good for tracking a white dot or something, yeah. Like a position sensitive detector, but it's not how the eye works at all. Anyway, what if you just sent brightness changes? In other words, changes in log intensity. That's a nice simple thing, right? So that's actually what the DVS does, like one of these cameras. It just sends brightness change events, just changes in this logarithmic output voltage. So just as a little exercise here, let's try to figure out what circuit it is that does this. So how can you detect changes in this logarithmic voltage here? Now you can memorize the log intensity and then look and see if the value changed away from the memorized value. And to save some time here, I'm just going to show you the circuit and then we'll try to figure it out, right? So what if you take this, you might need a buffer on it, like a unity gain buffer to isolate it, just like a source follower. And then what if you take a capacitor like this and another little one like this and just wrap an amplifier around it, an inverting amplifier. I draw it abstractly like a digital inverter, but it's just like this inverter on the left here. It's just like a high gain inverting amplifier. And then we'll put a switch across this. Okay, so what does that circuit do? Let's say this is C1 and this is C2. So if I connect that switch together, this reset switch, what happens to this inverter? If I just short that switch, well, then it comes to a state where the output voltage equals the input voltage, right? So it's going to be some balance voltage where the two voltages are the same. At that point, you know, it won't change its voltage, right? It'll come to some equal balance voltage. So that'll be like connected. Now, when I disconnect the switch, what happens? So now there's a charge Q that's stuck on this node here, right? Some charge Q. Now, what happens if I change this voltage here by delta V? Okay, from that point. So suppose the logarithmic output voltage changes by delta V. So what happens? So that puts a charge onto this capacitor, which is C1 delta V, right? That's how much charge gets stuck on that first capacitor. Okay, now, because of the feedback, this voltage here is not going to move. It'll be fixed. Like if the voltage here goes up, if this goes down, say this voltage goes down, what will happen to the output voltage? It'll go up, right? 8 times as much if this amplifier gain is A. And that'll tend to hold the input node of this amplifier fixed. So, but how much will this mode move? Well, if we put a charge Q, which is C1 delta V, 1, then how much will V2 be? It has to be that C2 delta V 2 equals minus C1 delta V1. So therefore, delta V2, in other words, the charge has to be the same, right? If the input puts a certain charge on, the output has to take it off. Otherwise, the voltage at the input of the amplifier is going to move. So delta V2 over delta V1 was minus C1 over C2. In other words, if this is 10 and this has 10 units of capacitance, this is 1, then the gain would be 10, minus 10. So it amplifies then the change in the log intensity by some ratio which is set by the capacitor ratio. That's called a switch capacitor amplifier. And it's a really nice way to build a precisely matched amplifier because the capacitors are going to really match well. You know, if you draw 2 capacitors, even a minimum size capacitor matches to better than 1%. So now you have the change in the output voltage here, right? And then how do you now detect that output voltage has changed by more than a threshold? Well, that's easy. You just put a couple of amplifiers there, like comparators, right? So you can put 2 comparators here. Let's just draw them like abstract circuits, like this. And then we'll put inside there a kind of a threshold function. So we'll make one have a positive threshold, and the other one have a negative threshold. And so this point right here in the middle is the reset point. So then the result of that will be that you'll get on if the output, if the logarithmic voltage goes up past the threshold, you get an on event. And if it goes down past the negative threshold, you'll get an off event. So that's how the pixel works. Every time there's an event that comes from the pixel, either you get a digital on or a digital off event. What happens is that this reset, causes the spike event to be sent out from the array. The location of the pixel is sent out from the array, and then that goes back and resets the amplifier. So that's what happens at up to about 10 million times a second in the output of the retina. Okay.
Excuse me.
Yeah.
Can you go
Are
briefly?
you guys chatting
again?
at me? There's people chatting at me and I don't see it.
No, not right now.
It's on Skype. Okay. Yeah.
Can you please go back and briefly on the off and how it works, the off comparator here?
Okay, so I have a better idea. So can you guys figure out now with the circuits that you know how to build these three components here? This one, and this one. Do you know how to build this amplifier? I can tell you it takes only 6 transistors. You can build this whole thing with 6 transistors. And you're already using the same circuit in this photoreceptor. You know how it is? You want to see the circuit? It's a really cool circuit.
Isn't it the same we use for the transimpedance amplifier?
It's just an inverting amplifier. They're all just 2 transistor inverting amplifiers.
Yeah.
So I'll show you the complete circuit, and it's a beautiful, simple circuit. I'll just draw it for you here, maybe in this color. So here is the input capacitor. Then you have a bias current down here. That's an N FET. A P FET here. Then you have another bias current here. Another P FET. One more bias current. One more P FET. And where's the feedback capacitor? There's a feedback capacitor here. And then there's a switch, which is a P FET, a little minimum size P FET. Let me draw it like this. That's C1, C2. And this is the differentiator current, and this is the on bias current, and this is the off bias current. On. And it's actually it's on and not off. It's like it's inverted actually. Yeah, you need one more inverter after this. That's the complete circuit. 7 transistors, 6 big transistors because you want them to match well. Because you want to have a decently matched threshold, and then 1 reset transistor. Hope that you guys can look at that later. See, that's really the same circuit. Nice and simple, huh? All kind of beautiful properties about this. In fact, the threshold voltage here is proportional to the thermal voltage if these currents have a constant ratio. In other words, if I on is 2 times bigger than ID, independent of temperature, then the threshold voltage is actually proportional to the thermal voltage. But this output voltage here is also proportional to the thermal voltage. So therefore, it turns out that the threshold for log intensity change is independent of temperature. You think about that a little bit. Anyway, let me do a demo of this retina. This is just the pixel core. I don't want to taking too long here. I'm going to fire up this camera. Move it over here. And I'll change the display to be this window, which is screen 3. Let me hide all of this. Okay, so I've got the camera right here. You see, it's like a USB camera. It's got a USB cable plugged into it. And here's the output. And so what you see, I don't know how many of you have seen this DVS camera. What you see is basically a kind of a window. I don't know how fast the update is there. But it's basically showing you spikes at a certain frame rate. Right. So if I just pause it here for a moment, maybe the best way is you can hear it, right? I can put my mouse on a particular pixel. Let's turn off the inertial measurement unit. And now you see that pixel 67163. Probably you don't hear this. Hold on a sec. I have to share the computer sound. Maybe you hear it through my speaker. Can you hear the spike sounds?
Not really.
Just barely, right? Let me stop the share and I'll share again with the 3 with the sound. Okay. Now you should hear the computer sound.
Now
Yeah, we can.
When I move my finger over the pixel, you can hear that particular pixel. And every time you see a white dot there or a dark dot, that means that in that time period, which is about 16 milliseconds here, that pixel has fired an on spike or black is an off spike. If it's gray, that means there was no spike from the pixel. Okay. So it's just like basically painting the events as white or black dots onto the screen. And so that's really nice. You know, it only sees movement, right? If I don't move the camera, nothing happens, but I think Chi-Chi's back there. I don't know if she's moving around. Now she stopped moving now. I have to move the camera to get an output. Beautiful thing about this is that it means that if you don't move, there's no output, right? If I stop moving, I just look at the blank wall, then the output event rate, especially if I turn off the frames, it has frame output too, but if I turn off the output of the frames, then it goes down to a few thousand events per second. This is kilo events per second. If I move the camera, it can go up to several million events per second. And so, you know, the data rate varies with the action in the scene, right? It's not like a normal camera where you just get a constant frame rate. And so it's nice for high speed stuff. I've done lots of high speed robotics with it. In fact, if I take something fast, like a deck of cards, and make a little riffle, I'll just do this demo here with this deck of cards. Let me focus it a bit. Let's see. Okay, so that's kind of in focus. And now I'm going to make a recording for a moment. Okay. Okay, I'll stop the recording. I'll save cards. And now let's play this back. Okay, so here it is in real time, right? But I can slow it down anytime I want because I've recorded, when I record the events, I record their times too. I can make it like really slow. I can make the frames like a few hundred microseconds long. So you kind of slow down the action afterward. So you can basically make you can basically deal with the data at any rate you like. So that's good for the high speed stuff. Now, there's another cool thing. I'll show you one more demo with this, which I think is quite cool. Do you have any question about it right now? Okay. So is it clear how the data is coming to the computer? What comes to the computer is just a list of numbers. It's the XY addresses plus one bit that says if it's brighter or darker, plus the timestamp of the event in microseconds. And so now you just have this, instead of having a list of pixels, you know, and their brightnesses in every frame, you just have this list of spikes coming at you. And your job is to now process that list of spikes, just like you're a piece of cortex, right? Processing the spikes from the eye. Only it's digital. It's just a digital representation.
In reality, are they a little bit batched because of the communications through the USB? Or?
Yeah, it's batched into packets. The packets come at a variable rate depending, I mean, if there's no action, the packets come, once every 100 milliseconds or so, very slowly. But if you really move rapidly across it, the packets come every 1/8 of a millisecond. So 8 packets per millisecond when you're using a USB high-speed interface. So the minimum reaction time, if you're using a high-speed USB interface, is about 1/8 of a millisecond, 125 microseconds. It's a lot. That's a pretty high-speed camera. Not super high speed, right? People are now building cameras with a frame rate of 1 billion frames per second, or even several billion frames per second. I can tell you about that some other time. But anyway, this lets you do a lot of high-speed robotics quite cheap because the list of spike events is much sparser than the list of pixels you have in a regular camera. Okay. You see me here? Do you see my video? Okay. There's an organ you have in your ear that you don't hear with. Do you know what it is? It's the thing that helps you balance. Have you heard of the vestibular canals? Yeah, these are like the inertial measurement unit of your head. They tell you how your head is rotating in space and also how your head is accelerating in space. And you know that every smartphone has one of these inertial measurement units on it too, right? Every smartphone has this accelerometer gyro chip in it that also gives the smartphone the sense of how it's moving in space, how it's rotating and how it's accelerating. And you can buy those chips for about $1 now, and they burn about 1 milliwatt. They're really cool. So you can basically build your own electronic vestibular system, right? That's what we did here on the retina. We put one of these IMUs on the back of the chip. Now, what is that good for? What can you do with that? Once you have this vestibular system built together with the camera, what'd you do with that? Well, you could electronically stabilize the output, de-rotate the picture. So let me show you. So imagine you're in a walking robot. What the hell is this? Okay, so imagine you're in a walking robot and you're outside and you're walking around like this. You're jiggling around. Maybe you're running actually, and so you're bouncing, right? It's a terribly chaotic view. You have this IMU. There's the IMU output. You see this pink thing here? That's the rotation sensor. It tells you how fast the camera is panning, tilting, and rolling in degrees per second. Now you can use that to compensate for all this motion of the camera. I'll show you how that works. I turn on this so-called steady cam. Basically, that takes the information, integrates it over time, and I'll show you the control here. So what you have to do in this steady cam, it's an algorithm that puts together the information from the IMU and the events, and you have to set the lens focal length. Do you see it here? To properly set the lens focal length. If I do that and turn it on, I should compensate for the motion properly. Let me try to show that to you. So this is with the IMU on. I don't think the lens is perfectly set. This is the IMU on and off. No, off. I'm not getting a good view. Let me just try to do it here. This is off. You see how it's moving around like crazy? And this is on. See how it compensates it? I'm moving the camera like wild now, but it just keeps it stable. Actually, your body does that too. When you walk and move around in the world, if you have taken too many antibiotics and you've killed off these hair cells in the vestibular system, your vision is not stable anymore. You'll just see a completely blurred world. You have to be sitting perfectly still if you have this damage to the vestibular system. And so your body is actually mechanically compensating the eye's motion all the time, while you're moving around the world. But because silicon is so fast and because these things are so fast, you can actually electronically stabilize by combining the IMU and the retina together. It's just one of the cool things that you can do for robotics. It's much easier to do it with these spikes than with frames because each spike can be compensated. In a frame, you have to derotate the whole picture. You can only do it at limited sample rate and so on. All right, do you guys have any question about that?
Yes, I have a question about the stabilization algorithm. So you use both the input of the camera of the lens and the IMU. And what kind of algorithm do you use to fuse the two? Is it a tracking system or?
Nothing so fancy. If you notice that it's nothing so fancy here. I'll show you the screen again. Now, do you see this red rectangle here? That's the transform that's computed by integrating this IMU signal over time. So the IMU tells you how you're rotating. So if I rotate the camera left or right, do you see that rectangle rotate? And now that gives us a translation vector and a rotation matrix that we can apply to every event address. So this rectangle is the integral of the rate gyro output. It's the integral of the derivative. So it's actually the position of the camera and angle. And now we can take that transform, which is just a rotation plus a translation. We can multiply the XY address of the pixel times the rotation matrix and add the translation to bring the event back to where it started, back to some arbitrary starting point. And we're just doing that event by event. You can read about it in a paper if you like. Does that make sense?
Yes.
So we take the XY addresses, we rotate it with this rotation matrix, a 2x2 rotation matrix, and then we add the translation, which is a vector of XY translation, and that compensates it. And just do that on each event as it comes out of the camera. And you can do that down at sub-millisecond timing resolution. Okay, so any other question?
Yes, I have another question. So did you just set what's the stabilized position at the beginning, or do you have to reset it every time?
It's high pass filtered right now. At the moment, it's just high pass filtered. So it only takes that high speed motions. You see when I moved it and then it kind of drifted back. Yeah, it just kind of removes the DC component of the rotation. So it's designed to to keep high speed motion stable. You know, these cheap IMUs that only cost $1, they have terrible mismatch. They're really bad quality. You know, every single one has a different offset. They'll tell you that the camera is rotating at, say, 1 degree per second, even when it's not moving at all. That's a big weakness of such cheap IMUs.
Mm-hmm.
So you have to high pass filter the output.
Okay, thanks.
So that's a nice thing for robotics, because you get nice sharp pictures, even if the camera is moving around wildly. Okay, so I don't want to take more time for that right now. Now, the next demonstration we have is from Min Liu, is one of our PhD students who's been now working with this retina to build a camera that completely computes optical flow within the camera. So optical flow is something that's computed by early visual cortex, right? You look in early visual cortex, you see almost all the cells are sensitive to the direction of motion. And it's also widely used. Min, maybe you tell about it. He's got his
yeah.
their environment.
Yeah, hi all, can you hear me?
Yeah.
Yeah, okay, so I'm Min. So I'm working on DOS, so that Toby just showed to you. I'm working on the optical flow. So I don't know if you guys know optical flow. Okay, so optical flow is, maybe I just give you a very brief introduction so you to know what it is.
I just say it in words, Min. It's just how much the pixels are moving, right? What's the rate of motion in the image?
Yeah, that's it. So what I'm doing is like I'm design this algorithm and also implement all this on the hardware. So let me show you my camera. So okay,
See your setup.
So it's I hope you can see it. So this is the camera. Can you see that? Yeah. Okay. Yes. So yeah. So let me also show my gear. I think I need to show my gear here. So share screen.
So actually, Min's camera takes the DVS, the dynamic vision sensor, and hooks it directly to a rather powerful FPGA.
Can
Like
you see
on the
all of them?
FPGA, yeah, on the FPGA, he computes the optical flow. Yeah,
Yeah,
it's a
okay.
bit blurry. It's not really in focus.
So that's my hand. If I use this one, I think you can see it's in focus.
Yeah, that's okay. Yeah.
Yeah. Okay. So let me give you a brief introduction. So this is the camera. This is the DVS, like Toby showed to you. So, but the difference between this camera and the standard camera is this camera can also calculate the optic flow, and in hardware. So this means no PC or no software is required. Okay. So let me show you like this one. So this is a checkerboard. So now this this optic flow is like it's disabled. Let me just enable it. Oh yeah, here. This. Okay. Okay, now let me. So if I move this one, so can you see the vectors? I think you can see it. So these vectors, yeah, this vectors means the output of the camera. So like I said, I do this on the hardware. So like if I just disable, because you see here's one option for this one. It's like enable hardware. So you can see it's very fast. Like you just move it it's like in real time. You can move it very, very fast. But if I use this, because also we have this software algorithm, if we disable this one, so now let me show you. You can see, so now all the algorithms are run on the PC, on the software. You see it's very, very slow. So our algorithm is a real-time hardware algorithm. So here. And also, yeah, and also another thing is like I want to show you this one, because this camera can be, I think you can see this. So there's one thing to configure this camera. So like currently, if you type H, it will show, okay, show play mode and change play mode. And if I just change this play mode, like, okay, change.
You're gonna load a file, right? You're gonna play a recording of event.
Yeah. Now I'm going to do that. So because now currently we are in the live mode. So if I go to playback mode, so you will see the pre-recorded file is in the camera. It will play back. So and you can see this the output. And also if I just go to another mode, so also go here. So there's one thing called like toggle ABM is our algorithm for the optic flow. So if I just, so can you see the difference?
Yeah.
Yeah, the difference. So the difference I think you can see is like there are more optic flow is output. So why there is more output flow? So because we have a corner detector before the optic flow. So it means that our algorithm now will first detect the corner and then we'll do the optic flow. So if we just use the original one, so then all the events will be processed, will be calculated. But if we just, so that's and also another thing is, I can show you this thing.
But do you know why it's useful to compute the optic flow only at the corners? Do you guys know about that? Anybody know why it's useful to compute at the corners and not at all the edges? Have you heard of the aperture problem? Yeah, so the problem with computing the optic flow at an edge, maybe you can illustrate it with your hand, man.
Okay, let me change it back to the live mode.
No, just show what's the problem of computing optic flow at an edge. Just with your hand.
I know.
Yeah.
So I think this one is better. So this one you see there are a lot of edges for this. for this checkerboard, you can see all these are the edges. So currently, I just calculated on this object on these corners. Why we do that? Because maybe this one is better to show you. So this optic flow, I think this one can see it very quickly. I think this Google wiki, you see, so like this bar, you see it like, okay, the moves like in this direction. That's true. We see like it seems like in this direction, but in fact it can be like this direction or any direction. I mean, if you imagine that, because we only see the local area of this bar, we don't see the whole bar, so we don't know the actual movement. We just know, okay.
You don't know the motion along the edge. The problem with this is that you don't know the component of the motion along the edge. You only know it perpendicular.
Yeah, that's true.
Yeah, but if you have a corner, you have a corner, it's unambiguous. Yeah,
that's true. So that's the reason why we do this on corners, like here, you see. So yeah. So in this mode, I just show you the corners. Can you see the red dots?
Huh?
So in this way, wait a minute. So you see the red dots are only on these corners. It's very clear.
So Min, what is this? So what's this optic flow good for?
So like Toby said, because our camera is very useful for the high-speed applications. So when you do robotics or do autonomous cars, so you need to like that will move in a very fast scene. So but you can't process for every event because they need the real time. So that means we can't process every event. We need to process only for the.
No, but you're not explaining what optic flow is good for in robotics.
So optic flow is like used for, like this example, you see. So if you have this optic flow, then if you also know the camera parameters, you know the distance for this car with the camera, then you can know how fast the car really moving. That's the very useful thing. So it means that if you know optic flow, then you also have like IMU or some kind of other depth camera, then you can reconstruct the environment, also estimate the speed of the camera itself. So, and also that's a very, very basic question for the robotics, because for robotics, they need to ask just two questions. Where am I and where I should go? So if you know the location, that means that you know where am I. So then you can do a lot of things. So that's very, very useful. I mean, for the SLAM optic flow, it's very useful for SLAM.
Good.
Yeah.
Thank you.
And
what's
do you have
SLAM?
any question? Yeah. What is SLAM? So SLAM is the simultaneous localization and mapping. I think if you do robotics, I think some of you should know that.
Okay, good.
Yeah. Do you have any questions?
L'ultima lezione di Anamorphic Engineering 1.
Giacomo, we hear you.
Sì.
Okay, so I hope you enjoyed that lovely demo. That's the world's first optical flow camera that uses this silicon retina. Now we're going to go on to, thank you, man. That was wonderful. Now we go on to Shiji. Yeah.
Can I have a quick question? I didn't really get why didn't every point individuate fire also among the corners? Like we saw in the one on the street when there was the car going around and it stopped, then it stopped and we were with the optical flow and everything. At some point, we didn't really see every point, every optical flow point. Or at least
it's always a threshold. There's always a threshold for detecting a corner, right?
Yeah, that's true.
Of course.
Okay.
Yeah. It's just a threshold. If it doesn't actually see a strong corner, it's not going to compute the flow there.
The algorithm, in fact, is called like FAST. If you're interested, you can just search FAST. It's a very popular algorithm for frame-based camera, but we just adapted to our event-based camera. So it's called FAST. You can, if you are interested, you can search that. They have a threshold, like Toby said. So this threshold will control the final output So if the threshold, so that will change the output. Yeah.
So the whole point of this corner detection is just to find points where you can reliably compute the flow without worrying about the aperture problem. You need nice high contrast. You need to be able to match across time, to compute the motion. And if the more reliable your corners are and the stronger the contrast is, the more reliably you can compute the flow there. Of course, you might miss some places you need the flow, right? It depends on the application.
Thank you.
No. By the way, the method that Min is using to compute the optical flow here is the same one that's used in every computer mouse. It's actually block matching. It matches the blocks of pixels that are sampled at variable rate across time. So anyway, he can tell you about more about that later. Now we have to go on to Shichi is going to take over to show you cochlea. Now you want to show okay, so let me
take this here, the whiteboard.
The whiteboard? Okay, so
Yeah.
we'll go over to the whiteboard. Now make a new picture. Yeah, go ahead. It's showing. It's showing it now.
How do I get a new one?
Go back here. Make a new one. You guys can see the whiteboard?
Yeah,
Yes.
Okay.
Yeah.
The
Yeah.
blackboard?
great. Okay, so before I show you how the cochlea work, before I show you what the cochlea puts out, let me just briefly describe what the electronic model of the cochlea is based on. So just start off with the biology, right? So remember with sounds coming in, you have your ear, and then there's a eardrum at the end of this ear canal. And then there's a set of 3 bones in the middle ear. And then going on to something that's shaped like a snail. And if you, sorry. So my drawing is not the greatest. Okay, so now if you take the snail-like thing and you unroll it, yeah, somehow, then what you see is in the from the eardrum to the 3 bones, the last bone will hit an opening in the cochlea, and what you see in here is a membrane that's shaped in a particular way in this chamber. Let me try and draw this. Right. So basically, it's very narrow at the beginning and then very wide at the end, and also it's thicker at the beginning and also thinner at the end. And so what this means is that if you play a particular frequency sound coming in, the place where the membrane will vibrate the most depends on the frequency of the sound. Okay, so what you can see then is if I play a particular sound, and now I'm going to draw the membrane in this direction.
Pushing the button. Yeah.
Okay, then basically you'll see some sort of vibration, and then at some place the energy will be dissipated very quickly, right? And then the membrane over here doesn't move.
Different color.
Okay, so there's some measurements that were done by a scientist back in the 60s on the dead cochlea. And so the idea here is to try and characterize what's the frequency response of the membrane at different places. And so now if I do this, then what you see is, if I plot this now in terms of frequency.
You're pushing the button.
Okay, then this is for higher frequencies. Let's see, now I have to flip it. It's tricky actually.
Doesn't matter. Just draw it. Transfer.
Okay, I'm just gonna draw it. Actually, it's kind of flipped the other way. So you're gonna see something like this, right? And this actually corresponds to here, and this corresponds to here, right? So basically, the front prefers high frequencies, and then the places at the end of the membrane prefers low frequencies, and it's somewhere between, say, 15 hertz to 20 kilohertz. And so if you, right, and this is the amount of displacement. So if you like, we're going to say it's a gain, some form of gain. And so if you look at something like this and you want to make an electronic model, what would you think about? In fact, yeah, I think you learned a particular circuit that could be useful, huh?
Bandpass filters.
Yeah, it's a bandpass filter, exactly. So that means that if you can make an electronic circuit that acts as a bandpass filter, then you've got the first stage of processing in the cochlea. And so the simplest circuit that you can make based on what you've already learned is you take the follower. So say this is the input coming from the microphone, right? And then you've got a capacitor. And what does this give you? Oh, here, sorry, you need something here. So
is that a high pass or a low pass?
It's a low pass.
It's a low pass. Okay, that's good. It's a low pass. And now I can make another one, right? So if I want something with a higher roll-off, because you can see here the roll-off here is kind of sharp, right? So probably want to have another stage that comes afterwards. Like, so again, there's a bias current. And so what order filter is this? You know?
Second order.
Yeah, it's a second order, right? So every time you add something with a capacitor, another stage you now with a second capacitor, then you get a second order filter. So which means the roll-off here will be 40 dB per decade, okay? And so if I now plot the frequency response of this, so you can do omega this time, right? Then what you get, it will be something that looks like this.
It's a low pass, no ring it.
Yeah. And this would be a 40 dB per decade because the second order filter. But you like to be preferential for some smaller set of frequencies, right? So which means that now you have to make the filter a little bit peaky. And so you like something that looks more like this, right? With a little bit of Q. So there's something called Q, and Q specifies the sharpness of the filter. And usually this is defined by your center frequency. So say FC here over delta F. And delta F basically is when the gain drops by 1/√2 of where you are at the peak. Okay?
So we just did Q for the photoreceptor. We defined Q as the number of cycles that it rings. That's actually the equivalent definition, right? FC over delta F is actually equivalent to the number of cycles of ringing if you work it out roughly. You would say, yeah.
Now to get this extra Q, what you can do is you can do this. You can put another amplifier in the backward stage. Okay.
Do you need another capacitor or not?
No, it's still second order. Okay.
So is that positive or negative feedback?
I need another current here.
How do we think about it? Is it positive or negative feedback?
Yeah, so be careful about this follower now because, well, it's not really a follower. So here the output is tied back to the plus side, right? So if you want negative feedback, you want it tied back to the negative input, but here it's tied back to positive input.
It's a little hard to think about, right?
Yeah.
If you think about it, though, you'll see that actually injects energy back. And it makes it resonant.
Yeah, it injects energy back into the output of the first filter, which is how you can get this little, extra gain, right? Where the Q now is greater than 1. Okay, I'll let you guys reason through this, if you like. And also you'll see that this Q is dependent on the ratio of the forward and the backwards current. That's what IF and IQ. Okay, that's how you can set the sharpness of the filter. And if you want to now to get this part over here, right, because this is a low pass filter, it's not a band pass filter yet. And so one easy way to do this is to take the output of the first stage and the output of the second stage, and then you go into yet another amplifier. Okay, so it's like a differentiator. And so what that would do is that would give you kind of a band pass filter look, okay? So it's not as sharp as what you get from the biology, but it's very expensive to create like a multi-order filter in silicon. Okay, so usually people tend to stay with second order sections or fourth order filter at the most. Okay, all right, so that means that we've got the front end of the cochlear channel. So now what do you do afterwards? So what's not shown here yet is that on top of this basilar membrane, so if I can draw it again, my basilar membrane, is that you get the cells, right? The outer hair cells and the inner hair cells, right? So that the inner hair cells, so if I take a particular stage over here, there will be 1 inner hair cell and 3 outer hair cells. Okay, 3 OHCs, outer hair cells, and 1 inner hair cell. And so this hair cells sit on the membrane because the membrane is vibrating and the cells have little hairs that stick into the fluid. So I didn't mention that it's fluid. in the chamber here. Then when the hairs move in one direction, it opens up ion channels. And so basically potassium flows in, it depolarizes the cell, and it causes it to release neurotransmitter, which stimulate some fibers. So there's some fibers that go out of the cochlea. And so now these are the ones that spike, okay? And basically this hair cells are acting like a rectifier, because when they do experiments, they found that if the hairs move in one direction, then the ion channels open up, but if it moves in the other direction, the ion channels are closed, right? And so we think of this like a rectifier. So in one way, you let the signal go through, and the other way, you don't let the signal go through, okay? And so what you can do now is you can put everything together.
Make a different color.
Okay, so now this is going to be one cochlear channel. But you're going to have the bandpass filter that models the basilar membrane. You're going to have a rectifier. Again, this is like a diode. You can think of that too. That models the inner hair cell. And then you're going to have integrated fire neuron circuit that models the auditory nerve fibers, ANF. Okay. So everything here is analog except towards the end, and then you're going to get this asynchronous spikes. And so now on this chip, we have 64 of these channels. So this is 1 up to 64. It's binaural, so it's so we have basically 2 ears going to each going to 64 channel, a 64 channel cochlear circuit. And what you should get when you do the measurements across the 64 channels against frequency, you're going to get something that looks like this. Okay. And there'll be 64 of this. And so on this particular cochlea, it spans somewhere between 20 hertz to 20 kilohertz. And what you're going to see on the output of the screen later on, so just like what you saw with the DVS, so here would be time.
The red and the green, even.
Yeah, so here would be the channels. So these are the low frequency channels. Sorry, these are the high frequency channels, and these are the low frequency channels. And there's 64 of this way. And then you're going to see spikes coming out. Yeah. And you're going to see a red spike because those are the spikes coming from one ear, and you're going to see green spikes, right, coming from the second ear, okay? And when they overlap in time, you're going to see them as yellow. All right, so you ready? Okay.
Now we have to share the other screen, right?
All right, so now we have to share the other screen.
Do you bias all the frequencies with the same weight? I guess I don't know how the human ear would work. I'm just wondering.
Good point.
You bias them.
How do you get the different frequencies?
Oh yeah, so to get the frequency, to get the different frequencies, what you do is you set the tau. So let me go back.
You change the bias current along the cascade.
So every channel will have like a different what do I call it? IF. Yeah, so every channel will have a different IF current. Yeah, and then you have to make sure that the IQ matches the IF so that you can get that same Q for everybody. Okay.
Clear.
All right. Any other questions?
So the channels on the right here are high frequency, have high bias current. The channels on the left with low frequency have low bias current, and you can make kind of a ladder. In fact, it's actually just a line of resistors, right? And it creates a linear voltage across the chip. And if you make a linear voltage and send it through a transistor, you get an exponential current, right? So the current changes exponentially from one side of the chip to the other.
So you can think of it like that. So here's, if I erase.
That's correct. Just like that.
Then here's one channel, here's the next channel. And what you do is you tap off the currents or the voltage, which is then sets a current, right?
There's a high voltage on one side and a low voltage on the other.
You have a V low and a V high over here, like that. Yeah. So you don't bring in 64 currents, that'd be too expensive, right? So you make a resistor divider and then you tap off the currents, voltages in this case. Okay. All right.
Hold on a second.
So now we switch over to screen, which is screen 3.
Can I ask a question too?
Sure.
That's a quick one. This is not my voice, right?
I can hear you.
Okay. Do we really need the resonance feedback, or could we just sharpen the bandpass?
Oh, I don't think I understand that. That's kind of the same thing. You're saying, do you need such a sharp filter? Is that what you're asking?
No, the resonance feedback for having the high Q, or rather for having the
to create
overshoot.
that higher Q. Yeah, right.
Yes, but do we need it, or couldn't we just use an actual bandpass?
You can design an actual bandpass circuit, but what's nice about having this feedback is that you can go and tune the Q of the filter. Because you like to show, like, okay, now I have a filter that's kind of broad in tuning, and then you set the biases so that you see a less selective filter, and then say, okay, let me turn it up so that the Q is higher, and then you see a sharper filter.
Okay.
Yeah. I mean, if you know exactly what you want, then you can just design it right in, right? Without, allowing for this variation. Okay, can you guys see the other screen?
Is it just cheaper than building a higher order filter to use the resonant feedback?
Is this cheaper? Yeah, it's cheaper. So it turns out that if you want to make something a high order filter, it's just very expensive. And so actually, in this case, so I didn't get into it.
You can't see the screen anymore.
Yeah, you can't see it anymore. But the so actually in the biological cochlea, it's a very sharp roll-off. I think it's over 200 dB per decade roll-off. So there's no way we're ever going to build this, right? And so Dick Lyon and Carver Mead, who built the first electronic model, what they did was they actually made a cascaded filter bank, right? So the output of the first filter feeds into the second filter, the output of the second filter feeds into the third filter. And because it's a cascaded architecture, when you look at the output of every subsequent cochlea stage, right, it is going to be much sharper, like an additional 40 dB per decade sharper than the output of the previous stage, right? And that's how you can get this really sharp roll-off as you go down this cascade. So actually the output of the cochlea that you're looking at is a cascaded filter bank architecture. It's not a parallel filter bank architecture, which is.
Why don't you show the output? Yeah.
Yeah. So can you see the output, guys? Here.
It's probably kind of slow. What is the frame rate for you guys? Is it like 1 hertz or a few hertz?
I don't know what that means for them.
I mean, is it, yeah.
So can you see the different channels here, right? So these are the high frequency channels going up to the low frequency channels. And then you see the red and green dots, right? Those are the spikes. And that's from the cochlea responding to my voice coming in. See, if I don't speak, then there are no spikes.
Oh.
Okay. And you see the red and the green spikes, and that's from the 2 ears. And then if the spikes are coincident, then they look yellow.
And, right? Do you see why it's curved over like that? What's the horizontal axis? Horizontal axis is time, right? So it's actually like about 30, 40 milliseconds each time from here to here. Did you see what it means when it bends over like this? What's going on there?
Because it's slower.
The lower frequencies are slower.
Yeah, and why is it slower?
Because it has to go farther in. It is for the lower frequencies, if I understood correctly, and those are like on the last part.
Yeah, it's right, because it's a cascaded filter bank, right? So you're right. So yeah, it go to the high frequency stages first, down to low frequencies. If you have a parallel filter bank architecture, then it wouldn't be, curved like that so much. Okay.
Want to show the 3D view of the events in space-time or something like that?
Are you going to play the piano?
First, yeah, maybe switch the view.
Toby wants to play the piano for you.
Oh, no, yeah, explain what's going on here and slow it down with the mouse wheel.
Okay.
Hello? Ooh, it's hard to see the different frequencies, but I hope you see the events now going away from you in time, right? Like they're red shifted as you go away from you.
Yeah, you guys understand the picture here? So it's kind of to give you this feeling of time going by, right? And so the channels are now laid out in this way. Okay, so
time goes away from you.
I cannot make a pure frequency sound. You see, there's a smaller band of channels that respond. And then when I speak, there are more channels that respond, right?
Okay. Now demonstration time.
Do you want to do this view or the other view?
The other view, maybe.
Yeah, this is too hard to see.
Now we take advantage of this piano we have here. Does it respond?
Yeah, it is responding.
I'll turn it up a little bit. You see the modulation?
Let me see.
I can't play the theme of 2001, but if I actually knew any music, I would play 2001.
So just think of this like the spikes are coming out from your cochlea now going up to your brain. Your brain has to make sense of it somehow. Okay, so I'm gonna now show you a video. So,
No, we go to Chang, right?
no, he's not there, so I'm gonna show a video.
Okay.
Okay. Let me see, how should I do this? I want to play this video.
Move that over to the screen on the left.
Yeah, okay, let me do this. Oh, it doesn't go.
You have to go lower, a bit lower. No, you move it down.
Okay, all right, and then now I share. How do I share? Oh, here we go, now it's here. Sorry, I think it's...
If you want sound, remember to share, yeah.
Oh, yeah, it's sharing. Okay, can you guys see this? Okay. All right. So what we did was we now took the output of the cochlea and then we fed it to a stage that creates spike count features, right? And then we drove a recurrent neural network with the spikes and we trained this network so that it would do a continuous speech recognition. So in this case, it's continuous digits like 0, 1, 2, 3, 4, 5, 6, 7, 8, 9, and 0. Okay. And so the way that we created the features is that we took the spikes coming out from a channel, and then we had a particular fixed bin time, and then we count the number of spikes that you have in that bin time. And across the 64 channels, we do this in the same bin time. And then now we feed this as a feature vector going to the network. And I have to show you a demo because...
Make it full screen.
Yeah. Because we didn't have time to get the thing up and going. But what you're going to see in the video here is here you have the cochlea. So here's the board. Where's your camera here? Let me move the camera to here. So you see the board over here? Yeah. So same board over here. It's going to an FPGA on which we've implemented this recurrent neural network. And then what you'll see on the screen first is the output from JAIR that you've seen before. So the different frequency channels over time. And what you see now on the right is an indication of which digit is being recognized. So it's blank, 1, 2, 3, 4, 5, 6, 7, 8, 9, 0, 0. And you should see the bars going up, right? So that says how confident the network is in that particular label. Okay, so just...
This is the setup of our spoken digit recognition demo using the cochlear board and our delta RNN accelerator. So this board is the cochlear board that is used to convert a microphone signal to spike events. And events is sent from the board to the computer using a USB cable. As you can see in the left window, you can see the plot of all the events. So the horizontal axis is the time axis, and the vertical axis is the channel axis. There are totally 64 channels, and each channel corresponds to a bandpass filter. The spike events also goes from the cochlear board to an FPGA board through a cable that transmits the spike events in the AER protocol. On this FPGA board, we implemented our delta RNN accelerator that is able to run a gated recurrent unit-based recurrent neural network to do the spoken digit recognition. And the recognized digits will show on this plot. So we have totally 12 labels, and the vertical axis shows the probability of the recognized digits. Now let me show you how the demo works. One, 2, 3, 4, 5, 6, 7, 8, 9, 0, 0, 0, 5, 6, 6, 7, 6, 5, 1, 2, 2, 3, 4, 5, 8, 9, 7, 6, 0, 0, 9, 9, 8, 7, 1, 4.
Okay, so you guys get the idea. Yeah. Yeah, so you'll notice that there's some digits that are actually quite hard, like 2, because some people can't say the t sound, so the cochlear doesn't hear it, right? If you don't say the t sound, and so then it's hard for the network to label the digit. Yeah. But you can actually make a very small ASIC out of these 2 pieces, right? So actually there's the ex-PhD student who designed this cochlear. He made a new cochlear classifier chip where he took some smaller set of the channels together with a classifier. Yeah, and then the whole thing runs like less than a nanowatt without all the extra stuff around it, right?
Microwatt.
Is it a
Nanowatt?
Well, the latest one, yeah, okay,
few.
maybe, yeah, you're right.
Few
No,
microwatts.
you're right. I think it's at least 100 nanowatts, but it's below a microwatt. Yeah. So you can actually get a very low power, cochlear classifier. if you like. Yes. Any more questions?
During the demo, we saw that he like called 6 3 times in a row, but the first 2 times the program got 9.
Yeah, that's the other thing is when you can't pronounce the S too well, right? Then the spikes are missing from the high frequency channels that let the network label it as a 6, right? So if it doesn't get those spikes, right, then it doesn't think it's a 6.
But how does 6 sound like 9?
Because
6 and
well,
9.
it's not like the sound sounds like, it's just which channels are activated, right? And the timing. So you shouldn't like say, well, the 6 doesn't sound like a 9, right? It's just whatever if it figures it's closest to it. Like if you're missing the S, then it's ich, if you like.
So if you ever tried to train such audio recognition network yourself, you see that it's much, much, much harder than you might think. It's actually really tricky to get it to work with all the echoes in the world, different sound levels. Oh boy, it's tough. Yeah. It'll bring you to your knees to get it to work. And you just take it for granted that it works now. But remember, that's the end of like 30 years of hard work in audio recognition that you can now get stuff that works.
Yeah, also when we did the data collection, because we had to collect, of course, new data sets, right? Because what you get from out there, it's mostly for just audio recordings. But if you only get spike recordings, you have to do it yourself. And most of the people that we recorded were Germans. So that means if you've got an accent or if you're more Asian, then it's more difficult for the network. Yeah.
Hard to generalize. Okay, so let's move on. Thanks, Shichi. Let's move on to the final demo, which is Dimitri. I hope Dimitri is standing by.
Yes, yep, I'm here. Can you hear me? Okay, can you hear me now? Hello.
You're good.
So my name is Dimitri. I'm a PhD student at NE, and I'm working with one of these chips. I can probably introduce you to the Dynaps chip first before I show the demo. I have a couple of slides showing what the chip does.
Maybe just to link it back to the class, it basically contains the silicon neuron and the DPI, the silicon synapses that we saw in the lectures, and more. So Dimitri can tell you the more details. But the circuits, it's related to the classes we did on the neuron and synapse circuits.
So I'll just, yeah. So this is basically a chip that contains 4,000 of these silicon neurons with 4 types of synapses, and they are organized over 16 different cores. And for every core, you have direct access to the biases. So I think you should have learned at least some of the aspects of how you would bias the neuron circuit. And here, essentially for the leaky integrated fire neuron, you have all these different controls for time constant, for DC input, for threshold, refractory period, and so on. And the idea of the chip is that you have full reconfigurability. So it first of all, it runs exactly in real time, so you can interface it with the DVS camera and or any other device that supports AR protocol, and then you can create and configure your network on this chip. And the access you have is with this program called Cortex Control for now, and now it's going to be even a more fancier version. And a good thing that it supports Python, so you can write just Python scripts which will process your spikes in different ways, and you can change connectivity. And there are some constraints to how exactly you can tune your network. But generally, you can connect any neuron of these 4,000 neurons to any other, and a lot of people at NE experiment with what kind of networks you can make work on Dynams. And the other way to get these to get data from the chip, you can also just measure it with the scope, so that you can actually see traces, and you can, if you have the scope, you can also extract it into Python code and do some loops and tricks to configure your actual time constants and weights. And then the example that I want to show here today is looks like this. It's supposed to be a winner-take-all, single winner-take-all structure where all the neurons are organized in a specific way. So they represent a basically ring structure or ring-like attractor. So every unit, which can be one neuron or several neurons, excites with decreasing weight its neighbors, and also sends output to a population of inhibitory neurons, which then inhibit all the neurons in the whole population. And what I'm trying to do here is to send a bump, or in fact 2 bumps, and try to get try to see how inhibition kills the weaker one. And on the chip perspective, this is how it looks like. So I can first shut off the unnecessary ones. So I can indicate the different connections or enabling different connections one by one sequentially. So basically, first we send the input, which are these 2 bumps. So the population we're going to be looking at is here. So these are just 16 single neurons on this particular core. So indexed from 1 to 16. And with Python, I configure the onboard FPGA generator to send Poisson spike trains with rates that correspond to this blue curve. So it's the input rates. And then here, every neuron, when it spikes, it illuminates here. And we also can record these events. So the chip never stops.
Why are there two, but Dimitri, why are there two red bumps? Two areas, like there's one on the left, there's one at the lower left, and there's one at the right.
Because I send 2 bumps currently. So
oh, I see. You're showing just one bump here in the plot neuron index.
I can just quickly get I can live.
Oh, I see. So you're sending more than one bump, not what you're showing there in the picture. Okay, that's clear.
Show the true plot, what is happening now here. So yeah, now the output rates are actually higher than input, which just depends on F curves of current set of biases. So I'm actually sending two bumps right now, and I would like to see how they interact when I start changing biases. So for now,
ask them a question. What's the question?
What's the question? Do you know what FF curves are?
So what's the experiment going to be here? You're sending in this input, which is which one? The blue bumps?
Yes, I'm
sending the blue bumps, and then you're going to turn on.
And I'm integrating spikes for 3 seconds in this case with this function get rates, and I get back. Where was it? This was the other one. And I'm getting back the orange rates, which is what's happening on the chip. And by changing the weights on the chip, I would like to have only one of these 2 bumps, of these 2 orange bumps, active when I enable the inhibition. Now it's turned off, just because I wanted to demonstrate what happens when I start enabling biases. So right now, only the input connections are active. So the connections from spike generator to population of excitatory neurons, which is this red one. When I start increasing local or lateral excitation, the activity starts to rise, and it floods the whole population. Then I go to another core and I enable this part. So the excitatory connections that go to the inhibitory population. And we should start seeing some neurons firing there. And as they start firing, the original excitatory population starts firing less. And then I can go back here and I start rising the inhibitory weight until I get back to the point where I have my activity under control. So now we have this balance of excitation versus inhibition.
It looks like they're synchronizing and there are these waves. If you just look at the blinking.
Yes, yeah. So this does depend. So up here we have the raster running as you saw with Cochlea demo.
And it's hard to see it though. We can't see any waves there, but I can see the waves going back and forth right between the 2 populations. It's obvious just by looking at the dynamics.
Yeah, and then it's the whole other story to balance time constants of excitation and inhibition, so that that you have your excitatory and inhibitory spikes resulting in smooth rates and in both populations actually, so that that there are no oscillations.
So you were going to show this magic thing where you were able to completely suppress one of them. I just leave the bigger one, right? That was the aim.
Hope. That's the hope. So, but of course I lost the perfect biases that do that right before the demo. But I'll just try to get it.
You go almost.
I'll just get more input here. A little bit more. I know this is embarrassing. Well, now it's just following, and I think I just had more. I had higher rates over here. But now, yes, excitation and inhibition are quite strong. So I just did it. I know. The good thing that with, I wonder which settings I have there. So with hard winner-take-all, where you only have one unit active, it's much easier because was there the winner-take-all lab in this course, because we had winner-take-all chips last year, but this year I don't know.
No, there was. We did have a lecture also on the winner-take-all circuit, but that was not using spiking neurons. It was the analog circuits, which is the hard winner-take-all.
So this one just follows the philosophy of the winner-take-all circuits, but is made with spiking neurons, and then here you have all these controls that you need to put in perfect balance to make it work properly.
Cool. Thank you. So we're out of time, Dimitri, for the class now.
I'm sorry, I cannot make it work it right now, but yeah, this is basically the idea is that with Python scripts and the accessibility, you can start writing some scripts that do this optimization and automate everything. And this is what I'm basically working on currently. But it's not at the level of populations. I'm configuring some more basic parameters.
Yeah, because these kind of recurrent computations are what characterizes Cortex. Unlike deep learning now, where basically everything is completely feed forward except during the learning by stochastic gradient descent or one of these other optimization methods. In Cortex, everything is going back and forth all the time, and everything is in balance. And the Cortex is doing its utmost to minimize the amount of spikes that it's making, right? Because that spikes are half the energy that your brain burns or to make spikes. And so the Cortex is just trying to minimize the number of spikes that it makes, trying to avoid overheating and consuming too many calories. And so this kind of feedback and this balance and trying to do useful things at that balance point is the crux of the kind of computations that Dimitri is trying to do on these spiking chips. And so you're at this sensitive balance point where you're most sensitive to changes in the input, but it's tough. stuff because you're doing a different style of computation. Is that a fair statement, Giacomo?
Absolutely, yeah.
No.
It's
But
more
if
like
you're
a
interested in this,
dynamical
another
system.
model of computation, right? That's closer to how the brain works, perhaps. Yeah.
Yeah, it's really using
That's not to worry.
dynamics and nonlinearities rather than feed forward and back prop. It's really a different.
Yeah. Okay, good. So I guess that concludes our demonstration for this thing. I'll be in contact with you guys about these exam preparation, Blackboard sessions. And otherwise, we wish you Merry Christmas and Happy Holidays and enjoy your peaceful studying time.